Hi everyone, welcome to another session of Sybeco. Today we are hosting Kort Driessen. Kort is in his final year of PhD with Chiara, Cirelli and Giulio Tononi at the University of Wisconsin-Madison. He's broadly interested in topics ranging from fundamental functions of sleep to the neural basis of consciousness. His work with Chiara and Giulio has so far focused on using a combination of electrophysiological, optical, molecular, and behavioral methods, and their goal is to investigate neural mechanisms underlying sleep need and function across spatiotemporal scales. Thank you, Kort, for accepting our invitation. Please go ahead.
Yes, thank you very much, and thanks very much for the invite here to Sybeco. I'll note that depending on how my presentation goes here for everyone, we do have the convenient option to rearrange the acronym to BPsycho, and I will also include some molecular data in a couple slides at least, so we might end up at BMO-psycho.
Just to be clear, I did think of BPsycho. I shared it with one of my senior colleagues, and he was like, I seriously object this name psycho, but that's funny. Yeah, but that was one of the options I was thinking about.
Yes. Yes. Yes.
Okay.
Anyway, regardless of where we went, where we end up with that, I will try to give everyone a sense of a project we've been working on for a while, a couple of years now, which should be published very soon, where we basically the goal of the project is to induce sleep-like brain activity in awake mice and see to what extent that activity per se can fulfill some sleep functions in those mice. So with that said, let's basically just get right into it. So the first thing that we need to start with that I want to explain is that sleep is regulated via these two fundamental processes. So we have both a sort of a circadian drive here that you can see with this yellow line. And basically that is constantly driving you to stay awake. So when you wake up in the morning, it goes up and up and up, and then sometime around midday it peaks and then falls off. In blue, we have the homeostatic sleep drive, and that's basically just going to build up and up and up the entire time you stay awake and then dissipate with sleep when you go to sleep. And so these times here, when you have very high sleep pressure and when your circadian drive to stay awake is falling off, that is typically when you'll go to sleep. And so this is sort of a very fundamental point about sleep, that it's homeostatically regulated, which effectively means that the less you've had of it, the more you need it. We also know that sleep is nearly universal across the entire animal kingdom. It's present in every animal species that has been carefully studied to date. And there are some very interesting ways that animals across the animal kingdom will try to get their sleep. And they will do all sorts of interesting things while they're sleeping and sleep while doing interesting things. So for example, reindeers will sleep while they eat. So you can see here that the non-REM trace looks like this. This is non-REM sleep. This is wake. And here, this is rumination, otherwise known as chewing of the cud, where the reindeer will actually manage to get sleep in as he's chewing his cud. Penguins will sleep while parenting, meaning while incubating their eggs. So penguins will sleep in these thousands of tiny micro sleeps that are just a second or two long. To remain vigilant, in order to remain vigilant for egg predators, basically. And my personal favorite example, the seal. Seals will dive down. So seals need to go down to the bottom of the ocean to get, off the shelf of the coast to get food. And so they will dive down and down and down. And they've figured out that as they're diving, they can orient their body such that they'll continue falling down, and they will get sleep in as they're diving down. And then when they hit the very bottom, they hit REM sleep, where the animal, and we and every animal, loses all muscle tone. And because that happens, they tip upside down and then have a REM sleep spiral where they tip upside down and spiral down, and then eventually wake up at the very bottom and go about what they went down there to do. And so this is just to say that animals will do whatever it takes basically to get sleep, and will fit sleep in wherever they need to. Sorry, I was just getting rid of the, there we go. Okay, there's also other interesting examples. I'm not exactly sure what to even call this, but this is a video of an octopus that is asleep, and you'll see here it's entering its form of active sleep, or what in mammals we might call REM sleep. And this is not, it's not exactly known what the function of this is, but you can see this sort of intense chromatic activity happening on its body. Other examples include, perhaps most relevant for this talk, unihemispheric sleep. And this was initially characterized and is maybe most well characterized in the cetaceans, which are things like dolphins and porpoises, beluga whales. And these are some classical recordings done initially in the Soviet Union, where you can see they had EEGs on both sides of the dolphin's head. And what they found was that as the dolphins are swimming along, one half of the brain will go into sleep. You can see that's the blue half here, the left half in this case, and the right half in this case. And the other half will remain basically awake. And the dolphins will coordinate this so that whichever side of the brain, so in this case, this would be his right eye, the other side of the brain would stay awake, and then these guys on the other side will do the opposite, so that he can have this eye awake on this side, and this one can have the other eye awake on the other side, and so they can sort of each watch their own side of the pod as they swim. So they actually will coordinate this unihemispheric sleep. This has also been found in birds that will manage to do it while they fly. It's also present in fur seals. This is all to say, all of this about sleep throughout evolution and throughout the animal kingdom is just to say that sleep, all of these things indicate that sleep is serving very likely a fundamental function that cannot be accomplished during wake. It just simply, whatever sleep is doing cannot be done with wake, and it cannot be recovered during even quiet wake, sitting there quietly with your eyes closed. Sleep is required. So this all begs the question then, what exactly is sleep? If animals are sleeping with one half of their brain at a time, what even is that? What does that exactly mean? And one way we can think about sleep is via the behavior of the whole organism. And so typically the things we look for are increased arousal thresholds, the fact that it's a reversible state of quiescence, the homeostatic regulation, as I mentioned at the start of the talk, a specific posture, and preferred sleeping location. And these are all things that hopefully are obvious to everyone that are sort of associated with sleep. But another way to think about sleep is via thinking more specifically about the spatiotemporal patterns of neuronal activity in the brain. And that is where I'll focus most of my talk for the rest of this presentation. And that's sort of what I want to give everyone an introduction to here, is how we think about brain activity during sleep. So just as a methods primer, when we're doing measurements in humans, we're typically measuring something like EEG, which is just for measuring field potentials, sticking electrodes on the outside of the skull and measuring the field potentials. In mice, we can implant a silicon probe, a silicon electrode down through the depth of the cortex and then record intracortically. So we can have an electrode, multiple sites on this probe and record all the way through the depth of the cortex. And then from that probe, we can record both local field potentials, which is just the extracellular potential on each electrode, as well as extract multi-unit spiking activity from those probes. And this is the model I'll use in the work I present here. And so that's most of the data that I will show. So then if we go and if we take one of these local field potential channels and just look at an example of what does this look like during wake, non-REM sleep, and REM sleep, you can see it here, and the differences should be pretty clear. So in wake, you can see that the field potential is relatively activated with high EMG. In REM, you can see there's also an activated field potential, just with very little muscle tone. And in non-REM, you see the very distinct appearance of these slow waves. And we know that if we quantify the amount of these slow waves, the amount in their amplitude, which we call slow wave activity is effectively just the delta band power, the spectral power in the low frequency range. We know that slow wave activity over time tracks the need for sleep. And so to see what I mean by that, take a look here. So this is plotting slow wave activity over time from a mouse as we do a sleep deprivation experiment. So this is, think of this time zero as the mouse's night. So he would be going to sleep here. We sleep deprive him for 5 hours just by introducing objects into his cage. He stays awake for 5 hours. Sleep need basically builds up and up and up as he spends time awake. And then you can see when he finally does go to sleep, indicated by the blue color, slow wave activity spikes and then eventually dissipates as he spends time asleep. So this is all to say that slow wave activity is a gold standard measure of sleep pressure. It is the way electrophysiologically we are able to measure how much sleep need an animal has. So slow wave activity is quite an important thing, it seems, or is at least tracking quite an important thing. So then we want to know what are the cellular mechanisms underlying slow wave activity. And to do that, we should go down to the single cell level. And so I'm going to show a couple classical recordings done by Marquez Steriade. These were in cats, but the principle should apply certainly across mammals, mice and humans, certainly. So let's take a look at that. If we record a single cortical neuron in vivo during sleep, let's start with wake. During wake, what we find is that during wake, the neuron is firing more or less tonically, basically all the time. Its membrane potential hovers around the resting potential, and it's more or less tonically firing action potentials. However, during non-REM sleep, you can see that the neuron becomes bistable. So what happens is its membrane potential alternates between these periods of desynchronized or depolarized firing, which we call up states, and hyperpolarized silence, which we call down states. And you can see here that the neuron is either in one of the two states. It's either spending time, its memory potential is either down or it's up. So it's bistable. Then if we go back to the network level, so now we have a probe down through the cortex and we're recording what I just showed you before. So the spiking activity of many neurons throughout the cortex and then a field potential. You can see the appearance of these slow waves once again. And further, you can see that the synchronous bistability is the cellular mechanism underlying this wave activity. So it's many neurons doing this exact thing all together. So to make this more precise, you can see that when many neurons all together enter a down state, we call that an off period. And that is the thing directly that underlies the slow waves. And when many neurons together all go into an up state, we call that an on period. So it is this synchronous flipping of on, off, that underlies the slow waves that we see in the field potential. Okay, so we further know that these on and off periods underlie slow wave activity. We also know that the off periods are themselves homeostatically regulated, meaning that they get longer and bigger when sleep pressure is higher. Yeah, is there a question?
Yeah, of course. So I'm curious first of all, thanks a lot for the great intro, because the majority of people here are not from sleep background.
Yeah.
So why is it that at the cellular level we see the neurons not firing, but at the LFP it seems like an increase in activity?
So at the right, so this is the spiking. Imagine we have a probe down through this cortical network in a sleeping mouse, right? This is the spiking activity of sort of every neuron that we can hear around the probe. And so you can basically see that the neurons are spiking, and then they all go silent. The positive deflection of the LFP, which I think is what you're getting at, that exactly. So that's just a convention. That's just an artifact. That's just a convention of how LFP versus EEGs are often plotted. This positive deflection in the LFP is neurons becoming silent. That is what a positive deflection.
Why does it's so weird. Why does it look like a positive deflection? Because for the rest, when they're firing also, we do have the positive deflection. It does go up, but less synchronous. Here, it seems like there is a large synchronous spike in activity, but then I'm not following. Why are we getting, neurons are not firing? Why do we see such a large?
So one thing you can think about is, I think this is mostly just a consequence of how people visualize and view the LFP and where things are referenced to. So this probe is referenced to the cerebellum. That's sort of one immediate thing, is that we're referencing the thing to a very different spot. And so sometimes people are referencing things differently and then plotting things differently. For example, just because of when you record the EEG, it is the other way. So when you're on the top of the skull, it does appear as a this decrease in activity does appear as a negative deflection. And so sometimes people will flip it to try and match that convention, and then you get this confusion. But I think it's clearer to plot it this way because you can think about this is outside of neurons, right? And so when all these neurons become silent, they're becoming hyperpolarized, meaning it's becoming more negative inside the neuron, thus more positive outside the neuron, which is how you get this positive deflection in the extracellular field potential. That's the idea, basically. But regardless, it's just to say that the result of this positive, whether you were to plot it one way or the other, the result of this thing that's happening is the synchronized silence of many cortical neurons.
Okay. Thanks.
Yeah, no problem. Okay, so because we know that these off periods themselves are homeostatically regulated, the idea is that maybe this on-off bistability, this on-off switching, is itself a critical mechanism for performing the function of sleep, as it's underlying the thing that seems to track sleep need the best. And if this idea is right, our thought was that we should be able to induce this on-off activity in the cortical networks of awake mice and do the job of sleep in that network while the mouse itself remains awake. And so that's what we try to do, and these are the experiments I'll describe. I'll try to describe for the rest of the talk. Okay, so the experiments we do basically are we implant dual silicon probes down into the cortex of the mouse. And the important thing to take from this slide is that one of the probes is just a plain linear electrode. So it's 16 channels down, and we're recording the entire mouse cortex. The other probe, which I'll call the optrode, has an optic fiber attached to it, which allows us to do optogenetics only on that side of the brain. That's how we're going to affect one half of the brain, one network, independent of the same network on the other side of the brain. So I'll refer to these as the optrode, which I'll constantly plot in blue, and the contralateral control probe, which I'll constantly plot in gray throughout this entire thing. And so here's what that looks like. So here you can see both probes, the optrode and the control probe. And this is just showing an example of when the animal is awake in non-REM sleep and in REM sleep. Okay, so the optogenetic methods we use are here. So we have basically two main optogenetic lines that we use to induce the off periods. And one of them, we express channel rhodopsin, which is an excitatory opsin, directly in somatostatin neurons. So we excite these somatostatin neurons, and these somatostatin neurons then turn all other neuron types off. And so you can see here, when we turn the laser on, the firing of all neurons recorded on our probe goes down only on the optrode side and not on the contralateral side of the brain. And then same thing with this other model, which is here what we're doing is expressing an inhibitory opsin directly in under the control of this CAMK2 alpha promoter. So you can think of we are expressing this directly in at least all pyramidal neurons of the cortex, and then directly inhibiting them via this channel.
Question.
Yeah.
Can you speak a bit about the areas you are doing this?
Yes, so we did this. The main way we thought about doing this, choosing the areas, was if this principle is right that we're sort of getting at, then it should be true basically everywhere, at least in the cortex. And so we picked sort of two basic areas that have been sort of well characterized before, a frontal area M2 and a parietal area S1. So we're basically all of our and we have all the experiments we confirmed in both of those areas, in both the frontal and parietal area.
Parietal, you mean PPC or you mean somatosensory?
Parietal, I mean somatosensory.
Somatosensory. Why would you call somatosensory PPC? They're different.
Yeah, that's right. I don't mean posterior parietal cortex proper. I just mean parietal cortex as in not the front. But yes,
I see.
It's a good point.
And then the other question, mostly to shut down the cortex, people use activation of PV neurons. So somatostatin is not really used as a way to inhibit. So I was curious.
That's right. Yes. So the reason we chose somatostatin is a very specific reason, which is you can see here. And if you want more details, the best reference is Funk here from 2017. That's a paper from our lab. And basically, the reason we chose somatostatin neurons is because we have very strong reasons to think that somatostatin neurons are involved in the natural generation of off periods during non-REM sleep. And so if you look at the field potentials of what happens when we pulse the laser, as shown here by blue for these two models, in the somatostatin case, we get this big positive going deflection in the LFP, which looks just like non-REM sleep, actually, natural non-REM sleep. When we pulse here in the other model, we get this big negative deflection in the field potential. So it's working via a different mechanism. And we have reason to think that this one, meaning the direct activation of somatostatin neurons, is the thing that could be at least partially involved in initiating off periods naturally in non-REM sleep. And these neurons are actually known to inhibit every other subtype. So they're sort of a good candidate.
In the somatostatin row of plots, if we look at the one on the right, that upthroat, there we see inhibition. But you're saying if we really zoom in the first 80 milliseconds, there is an excitation, and that's what you should. So I'm comparing that with if you move your cursor to the right, where you have the upthroat, the blue. Yeah, this versus the one on the left. So they're both the same condition, but one looks like positive deflection, the other is negative. I'm just comparing.
Do you mean here versus here?
No. Somatostatin
here versus
upthroat. Yeah, the upthroat. No, the two upthroats.
Right, this one versus this one?
No. That compared to the one all the way to the right. Are they not the same condition? Yeah.
Yeah, these are so this is showing the same data. This is just the field potential, and then this is the spiking activity.
Okay, I see. So again, the same thing. Right.
Yep.
Right.
So it's just saying the thing you can take is that we get this positive deflection, just like you would see in an off period in non-REM sleep, and we do indeed shut off the neurons. And then the other side of the cortex is relatively basically unaffected.
But again, this goes back to my original question, and I don't want to dwell on it because I want to listen to the rest of your talk. But at the bottom, even though we see inhibition at the spiking level, still the same reference electrode, same everything at the LFP is giving you negative deflection. So it is really a question that why, then it's not about the reference electrode here.
No, right, that's correct.
Yeah. So
these are referenced to the same exact spot, and they're implanted in the same area. So this is a question about, this is a deeper question about basically the role of
LFP is picking up here and how these are different in terms of their extracellular currents, because that should
exactly. So to give you the short answer, and we can talk more about this at the end, maybe I have more plots and things, but basically the thing you can think about is I can give you additional references. The one interesting reference would be Hannah Alfonso from a couple of years ago, a paper in Nature Neuroscience. Basically, intracellular chloride concentration seems to be regulated by sleep pressure. So as you spend time awake, the reversal potential of GABA A receptors can actually change. We think what's going on, these STGTACR1, this means the relevant acronym here is ACR. It's an anion specific channel rhodopsin, which means chloride effectively in this case. Yeah. Exactly. So we think what could be going on here actually is that you have this interesting thing happening with chloride where you have a lot of chloride intracellularly, and then when you open these channels, it comes out, hence the negative deflection, but you still get inhibition because the neuron gets forced to the chloride reversal. Could be one idea, but it's a difficult thing. I don't really have a full complete sort of answer to this, other than to say we think that this is the more physiological representative and sort of useful model to use.
Okay. Cool.
Okay, so then this is the basic design of the experiment. So again, we're going to sleep deprive mice for 5 hours. As you can see here, we're plotting slow wave activity on the y-axis again, and then this gray line is just showing the slow wave activity of just the contralateral control probe. So we sleep deprive the mouse for 5 hours. He goes to sleep, and then we measure slow wave activity spikes, and then we measure a number of things in the 1 hour of non-REM recovery sleep following sleep deprivation. So I'll refer to this as recovery sleep or the non-REM rebound, right? The rebound in slow wave activity, which then dissipates as the mouse spends time asleep. So this is just to show you in baseline conditions, the two probes are extremely matched. So when we don't do any manipulation, the two probes go basically perfectly together. The two sides of the brain, the same cortical network mirrored on both sides of the brain is basically, they're very synchronized with each other. And so the idea is if we can induce 30 minutes of on-off period induction at the end of the sleep deprivation when a lot of sleep pressure has built up, perhaps we can effectively pull that rebound, that recovery sleep forward in time for one half of the brain only. And then when the mouse actually goes to sleep, we should see lower sleep need on that side of the brain relative to the unaffected control side of the brain. It's like this side has already started its rebound. Okay, so that is what we do. And basically, let me show you what that looks like. I want to show some raw data. So this is just going to show you baseline non-REM sleep when nothing is happening. So here you can see both sides of the brain, the control probe and the optrode. And basically, you can see the occurrence of these off periods, hopefully fairly obviously, associated with these big slow waves, as we were discussing before. And you can see that there's a number of them. And more importantly, you can see that on both probes, they appear to be relatively synchronized, quite synchronized, actually. And so when we overlay these things on top of each other, you can see that we have a lot of synchrony between the right and the left side. And these off periods are occurring basically simultaneously on both probes. Now, when we do this manipulation, here's what this looks like. So now the animal is awake and moving at this point when I show this data. And you can see that while the contralateral control side is basically has an activated LFP and more or less tonic firing, on the optrode side, when we pulse the laser, we get this big positive going slow wave. And we see we basically induce an off period in the cortical network. And that happens every time we are pulsing this laser. And so here now, if we overlay these two on each other, you can see that we have done this optogenetics mediated dissociation between the two networks, which hopefully is clearer to see here. So you can see indeed that on the optrode side, we've induced this non-REM-like on-off bistability, while the contralateral control side basically continues its typical wake-like activity patterns. Okay, so then we'll see if this video plays. Oh yeah.
Tell me if I should wait. Was that then somatostatin? Because there was the
yes, that was just showing exactly. That was just showing an example from the somatostatin animal. Yep.
I think you're going to talk about this, but I'm super curious about what the mouse does if one side is sleeping, the other is not. So do you see? Yeah.
So here's a video of the mouse as we do this. Okay, so here the mouse is basically awake and moving around. We're not doing any manipulation here. This is just the mouse in his cage running around playing with toys and objects that we give him. This is in non-REM sleep, and you can see the off periods are marked in red here, and he's obviously just asleep in his nest. And then here you'll see this next one, you will see the induction, where each one of these is a pulse of the laser. We're inducing these off periods, and the mouse basically walks around his cage, you can see down here, and appears completely unaffected by everything we could tell. We did a number of behavioral analyses, analyses of the EMG, of video, and we could not see any effect on behavior during this stimulation. The mouse appears to walk around and operate as normal.
So by inducing sleep in one hemisphere, if I understand
in one local cortical network of one hemisphere.
One local cortical network, but one side unilaterally.
Yep.
You manage to wake up the mouse. So the mouse wakes up and start. You manage to. Okay, so one side in one area is sleeping. Did anything happen to the mouse? Mouse feels less awake.
No. So here you saw this video where the mouse is basically running around his cage and playing with these toys we've put in there during the sleep deprivation. And here you can see each one of these arrows is us inducing an off period on one side of the brain.
Right.
Us doing that in one local cortical network, us doing that does not affect the mouse's behavior. He continues to run around his cage and drag stuff all around, as if we weren't doing it.
And this is towards the end of the wake cycle.
Towards the end of the 5-hour sleep deprivation.
The sleep deprivation.
Yes. So if you remember here, right, we're doing a 5-hour sleep deprivation. Where we then try to do this induction at the very end of the sleep deprivation, right before the mouse goes to sleep. So we're doing this here, we're trying to induce this slow wave activity at the end when the mouse itself remains awake, running around.
Right.
And then when he goes to sleep, we should see on that side of the brain that we've released some of that sleep pressure while the mouse was awake.
And do they wake up earlier because one side wakes up earlier, basically
Do
because
the
the mice?
no, so we
after,
right, yeah. I can show you at the end some additional figures. I also have unpublished stuff I can sort of show. But no, we don't see any effect on sleep architecture, latency, or anything like that.
okay.
Right, so then, yes, exactly, right here. So this is just an overview of the experiment. So you can see here, this is the hypnogram down below. The mouse is awake this entire time. Obviously, slow wave activity is very low on both sides of the brain. The idea is that we can pull the sleep rebound forward and then affect slow wave activity only on that side of the brain in subsequent sleep. And indeed, that is what we do. So now this is showing real data from the actual experiment. You can see the slow wave activity measure on both sides of the brain from both probes is quite tightly correlated the entire time until we start this procedure, at which point the slow wave activity spikes up only on the optrode side, declines a bit as we continue this on-off induction protocol. And then when the mouse goes to sleep, indeed, the control side, which was unaffected, spikes up. And then the optrode side, where we did the manipulation, is lower. It's as if we've released some of the sleep pressure, some of this rebound during wake. It's as if we've pulled the sleep rebound forward into wake only for that local network. And indeed, we see this across every single mouse we tried. So across every single mouse, we see a decrease in slow wave activity on the optrode side relative to the contralateral control side. And we see that it's not some broad spectrum weird thing we're doing, that it is specific to the low frequencies where slow wave activity is measured. So then the next question is, how are we actually doing that? What is the mechanism of that? And if we look at the raw traces, we can maybe get a bit of a clue. But our first idea was actually that we were affecting the architecture of sleep in some fundamental way, meaning completely eliminating these off periods or changing their duration or something. And that is basically not the case at all. We don't affect the frequency of these off periods. We don't affect their duration at all. So the general architecture of the activity patterns in sleep stays the same. So the question is, how are we getting reduced slow wave activity? And if we look at the raw traces, so now on the left side here for both mouse lines, you can see what if we overlay one channel from one side of the brain onto the same channel from the other side, you can see that during baseline sleep, when we're not doing anything, the mouse is just asleep, they are, for every slow wave, the two sides of the brain are very tightly correlated. They're almost perfect matches of each other. In recovery sleep, after our manipulation, it's especially clear in the SOM mice, you can see, indeed, we've reduced the amplitude of these slow waves. It appears that we've reduced the amplitude, at least looking at the raw data. And so this basically gave us the idea that maybe it's not the amount or number or duration of these off periods that we're changing, but the synchrony with which the network enters and exits these on and off periods. And so that is what we tried to look at. So we basically detected these off periods, and then you can look at the field potential, right? So when you have an off period, you have this big positive going slow wave, and you can look at the off period initiation. So this initial part of the slope here, right? You can look at when the off period terminates or when the on period starts. So this negative going slope here, and you can look at the peak. And the idea is that if synchrony is higher, you should have a steeper slope here, a steeper slope here, and a higher peak. And indeed, so now this is just in baseline again, you can see that the two sides of the brain are quite tightly correlated to each other, the same thing I've been saying multiple times. And then when we go into recovery sleep, indeed, we see this across all measures, that the slope is decreased when the off period is initiated. The slope is decreased when the off period is terminated, when the on period starts, and the peak is on average lower. And again, this occurs across nearly all mice that we tested. We also were able to look at spike timing measures of neuronal synchrony. So here, for these analyses, we computed what's called the spike time tiling coefficient. And basically all that does is for every possible pair of channels of multi-unit activity we have, we can say for channel A, or for spike train A, what proportion of spikes in that train have a partner in the other train within, let's say, plus or minus 5 milliseconds is the typical way you look at it. And if all of the spikes from one train have a partner very close to it in another channel or train, you can say there's high synchrony there. Whereas if most of the spikes in this train are happening, and then on the other spike train, there's no sort of closely associated spike, that's sort of a low synchrony condition. And so that's what we computed. And you compute this between two channels, basically. And so we computed that across all possible channel pairs for these mice. And you can see basically these plots are showing, the way to think about this is that blue means that synchrony was reduced on the optrode relative to the contralateral control. Red would mean synchrony was increased, which would be odd, and gray would mean there was sort of no change. And so in the baseline condition, once again, the two sides that indicate 0 here, 0 means there's no difference between the two probes. When we go then into the rebound sleep, you can see that across nearly all of these channel pairs, synchrony is reduced on the optrode relative to the contralateral control probe. And then when you average at the subject level, you can see that pretty clearly. Okay, so we were able to do this to reduce slow wave activity, LFP, and spiking measures of synchrony. And so we have that effect, and that's very clear. One objection to these results that I've just showed you could be that perhaps this on-off activity that we're doing that is meant to mimic non-REM sleep is not itself what actually matters, but just giving the neurons a break from the high firing rates they're usually sustaining during wake. In other words, if we tonically reduced firing rate in some way without having the on and off, couldn't that do the job just as well, basically? That would be one potential objection.
Kort, question.
Yeah.
So if we can pause a little bit, there's a lot of information, and I think you've gone through some of the questions I'm going to ask, but I want to make sure I'm following. So the reason this induction, this activation of whether the GTACR or the somatostatin neurons, the reason that reduces sleep pressure is basically because you make one hemisphere, I know one cortical area in that hemisphere sleep earlier. So it's like that sleeps earlier. So then when you release that and mouse goes to normal sleep, then it's like mouse has already slept a little bit. So that's why. However, there was no reflection of that reduced sleep pressure in the mouse's behavior. So behaviorally, mouse just had his normal hours of sleep and wakefulness, right?
Correct.
So how did you exactly conclude that this reduces sleep pressure? It was based on the synchrony measure?
It's based mostly here on the slow wave activity, which is the gold standard measure of sleep pressure. A more precise term, so you're getting at something that's sort of very true. A more precise term here would be local sleep pressure. And we know that sort of sleep pressure can accumulate naturally and dissipate sort of asynchronously or heterogeneously between cortical areas. So for example, there's studies that if you or a mouse are using, let's say, if you're a human learning a very specific motor task, when you go to sleep, your motor cortex is going to have higher sleep pressure than the rest of you, even though you yourself may not sleep more or less or earlier or later. And so sleep pressure itself, sleep need, can build up differentially between areas based on
right.
What's going on, how they're being used, the activity in those areas.
And the sleep pressure plots on the left, those are measured, it says 1 hour recovery. So that those are measured during the first hour of recovery sleep.
Exactly.
And then because the magnitude is lower on the opto side, you say that sleep pressure then must be lower.
Yes, in that network compared to the same unaffected network on the
Yes.
contralateral side, yeah.
Right. So basically you're reading out the magnitude of the slow wave as the sleep pressure.
Correct. Yeah. Yes, that's basically right. Slow wave activity, to be very specific, is delta band power, which encompasses both the magnitude and the sort of frequency. But yeah, effectively it's a measure of how much and how big, how many and how big the slow waves are.
Okay, thanks.
Okay, so then, right, as I was saying, for all of these results that we have so far, basically, the question is, can we sort of answer this objection that it's not that we've induced this non-REM-like on-off activity that matters, but just that we've reduced firing rate. And if we just took firing rate down without this on-off, that would do the trick. And so we did it.
Question, Kort. Did you have to do this manipulation at the end of the sleep deprivation, or if you were doing it earlier, that would still affect the magnitude of the slow waves during the recovery sleep?
So the reason we did it at the end of the sleep deprivation is because if we think that this manipulation can release sleep pressure, there must be some sleep pressure to release. You see what I mean? And so if you were to do this at a point where the mouse is not tired, it may have an effect, but it's harder to interpret in some ways. And so we thought this was just the...
But it could be like napping during the day.
Exactly. Yeah, that's right. Yeah, that's an experiment I would actually really like to do is a nap version of this. So imagine if we did the manipulation 3 hours in, right, and then continued it, and then he went to sleep, it'd be like the mouse had a nap, effectively. So that's an experiment that, yeah, unconstrained by sort of time and stuff, I would definitely do. Okay, so then here we use this model to try and address this question about tonic firing rate reduction. And this is using halorhodopsin now, which is a pump, which is sort of more suited to this type of manipulation. And so this works via an inward chloride pump. And so we're expressing this inward chloride pump in all, at least in all pyramidal neurons throughout the cortex. And when we turn the light on, we pump chloride in and inhibit neurons. That's basically the idea. And then we do this same exact experimental design, except during the final 30 minutes, we don't do on-off period induction, but we do a tonic inhibition of firing rate, meaning we turn the laser on and we leave it on, and then we are able to tonically reduce the firing rate, so bring down the spiking activity of neurons, but without inducing this very distinct on-off, up-down activity. And you can see that the result of that is when we do the manipulation, we do not see an increase in slow wave activity during wake when we're doing the manipulation. And then the idea is that when the mouse goes to sleep, if firing rate reduction is what matters, you should see lower sleep pressure. And if it doesn't matter and this on-off bistable activity really is critical, you should see the two sides of the brain the same. So indeed, we find when we do this that we can reduce firing rate. So in all the mice we tested, we do reduce the firing rate when we do the manipulation to the same amount or a greater amount as when we do the off-period induction. However, we do not induce off-period, so this is showing off-period frequency, and we do not induce slow wave activity, shown here. Then when we go to the rebound, we indeed find that there is no difference in slow wave activity between the two sides of the brain. And then when we do direct comparisons between all three lines, experiments that we did in this sort of paper, we see that if we look at the overall firing rate of reduction during the optogenetic manipulation, we see that the overall firing rate over those 30 minutes, right, was reduced in all three models as expected, but that firing rate reduction was not different between the models. So we basically reduced firing rate the same amount in all of these experiments. However, if we then look at the off-period frequency, we see that here, so 1 would be a frequency equivalent to non-REM sleep here. In both of the off-induction experiments, we induce a lot of off-periods, similar to a rate seen in non-REM sleep. And in the halorhodopsin experiments, we effectively induce none. And then indeed, when we go to slow wave activity and see what was able to reduce sleep pressure, only the off-induction experiments were able to do that and not the tonic inhibition.
So the difference in apart from one is halo, the other is the other ones are other things, was in the duration of opto. Could you remind me if you could go back?
The difference is best seen here, right here.
No. The difference in the opto protocol, I mean. So
yes.
So in one case you did it tonically.
In one case, we're inducing this on-off activity with duration and frequency statistics similar to what is seen in natural non-REM sleep. In this case, rather than doing that, so rather than using 100 millisecond pulses or something, we're turning the laser on for 20 seconds at a time continuously.
But then you also are changing your opsin. So if you were sticking to the same opsins and just changing the duration of optogenetics instead of doing on-off, would you see the same?
So we have a figure in, we have another sort of supplemental figure in the paper showing exactly what you just sort of described right there, where we try the tonic inhibition with the same somatostatin model. And we see roughly the same result. The issue is that it was much harder, we found, to get this tonic inhibition with the somatostatin model. It's with these ion channels, it's much harder to drive them for such a long time, continuously like that. And so
then the on-off in the previous protocol, if you could go back a few slides so I can see it, where you show it for the somatostatin, and there, it's not like you have closed loop. Is it like you're monitoring the activity and then you use that to decide on the optogenetic parameters? It's not closed loop optogenetic.
No, it's not closed loop. This is just we're just giving the pulses at a specific frequency with a specific duration. So we start with the duration being longer because off periods are typically longer at the very start of recovery sleep, and then we eventually reduce the duration of those pulses. But it's not that we're doing anything closed loop, and this is probably the main thing I would like to change about if I went back, the easiest thing I would redo is just take the exact statistical distribution of off periods from natural non-REM sleep and then replay that exact thing into the mouse's brain on a per mouse basis. But anyway, we did sort of an approximation of that.
Yeah.
Right. So tonic cannot do the trick. That's the idea here. Okay, so then in some sense, though, reducing slow wave activity or reducing sleep need is not really the function of sleep per se. The function of sleep is to perform some critical function, which then shows up as reduced sleep need. And so one project of our lab over many, many years has been developing a hypothesis for the fundamental function of sleep called the synaptic homeostasis hypothesis, or SHI here. And basically what SHI says is that the fundamental function of sleep must be a period of net synaptic weakening. So basically, when you spend time awake and you're interacting with the environment and you have ongoing learning, it must be the case that synaptic strength, at least through the thalamocortical system, is constantly building up and up and up. And then what SHI says is that the function of sleep must be to provide a time when synaptic strength can renormalize, when you can have a net weakening occur, so that you can basically prevent saturation, do a reset of a sort, and then continue to learn the next day. And there is now very extensive evidence for this hypothesis across many species and many different experimental methodologies. So just to give you a few, we've done electron microscopy studies where we directly go in and measure the size of the synapses themselves at the axon-spine interface, and we find that after sleep they actually shrink. We've used EEG methods with TMS in humans, looking at evoked potentials. And we've done molecular experiments where we can actually go in, take the tissue from a mouse after they've either been awake or asleep, extract the synaptoneurosome, so extract the proteins just from the synapses themselves, and then look at what's called GluA1, which is a subunit of an AMPA receptor subunit, basically, and which is known to be involved in synaptic plasticity, and which is known importantly to be regulated by sleep and wake. So after spending time awake, the amount of this GluA1, which you can think of as being like AMPA receptors, right? The amount of AMPA receptors goes up after wake and then goes down after sleep. And this is just showing the phosphorylation of that subunit at this serine 845. So normally this is what happens. When you spend time awake, you get more of this builds up, and then when you spend time asleep, it's able to renormalize down. And that is the method that we are going to try and use with this protocol I've described to you to see if we can actually perform what we think is a very fundamental function of sleep without the mouse actually going to sleep. And so this is how we do this experiment. So it's a very similar thing. We implant EEGs on both sides of the brain, but on one side only. We implant 4 optic fibers, so we can do optogenetics. The reason we implant 4 instead of 1 is just so we can affect a much bigger area of tissue, so that we can take that tissue and then do these quantitative immunoblotting molecular experiments on them. So we then do the same exact experimental design where we sleep deprive the animal for 5 hours, and then we do this off-period induction using the same exact protocol as before, only during the final 30 minutes. And then when the experiment is done, the mouse is not allowed to sleep. The mouse is immediately sacrificed, at which point we collect both sides of the brain, the control side where nothing happened and the off induction side, and then we measure the amount of this GluA1 in the synaptoneurosomes. And so here, this is showing the EEG. You can see that indeed we induce slow wave activity on the opto side, not on the contralateral control side. And then in the control mice, the wild type mice, we don't, nothing happens. And indeed, you can see that relative to the contralateral control side on the left here, the off induction side shows a large decrease in GluA1 for both models, and the control mice show nothing. And this reduction of about 30% is roughly on par with the magnitude you see following actual sleep. And this also occurs for the phosphorylation of these subunits at serine 845. So that the next sort of obvious question here is that we know another function of sleep, certainly in some ways, is memory consolidation. And we know from previous work that we've done using microscopy methods that if you look at the amount of what I'll call downscaling, reduction of synaptic strength that occurs, so this would be to the left would be more downscaling, that is correlated with performance on a task the next day in mice. This isn't a motor task in mice. So we basically have reason to think that the amount of downscaling might actually be correlated with performance, meaning consolidation of learning. And so that is the next thing we're going to try to do is can we use a task where we can induce these on and off periods and then restore memory consolidation, which would otherwise be lost to sleep deprivation.
Question, Kort.
Yeah.
I don't know if you guys did this experiment, but with the tonic inhibition, the one that you showed does not affect the magnitude of the slow waves. Do you see a change in the synaptic markers?
So that is not in this paper. We've been doing follow-up experiments of many kinds, and basically they're not exactly, everything's not done yet, et cetera, but the direction is that no, we do not see this change when it's tonic.
You mean like based on the previous experiments in your lab?
Yeah, based on experiments that are sort of ongoing currently.
Right. So,
yeah.
And then this is a discussion point. We can come back to it at the end of your talk. I'm very, very curious that what is about this, because tonic doesn't do it. We need the on, so the off is not sufficient. And I'm very curious what is about that induces this.
Me too.
Homeostasis. Yeah.
Yes, me as well.
I'm curious if you have any.
We can talk about that at the end. Yeah.
Sounds good.
Okay, so then let's go to right. So here the point is that we are basically asking whether this protocol, this on-off induction, can restore memory consolidation. And so here we have a task where we now implant bilateral optic fibers, so we can induce this activity bilaterally, and we then put these mice through a task of memory consolidation, which is known as floor texture recognition. So the idea is this. The idea is that the mouse goes into this chamber and everything about the chamber, the two sides of the chamber, it's completely identical. And he's in there for 15 minutes. He's allowed to explore. He then goes back to his home cage where the mouse is either allowed to sleep in his home cage, he is sleep deprived, or he is sleep deprived, but we also do off-period induction during the sleep deprivation. Then he just is left alone for the next 23 hours, and then 24 hours after the initial acquisition phase, we do what's called the recall or the test phase, where the texture of one half of the floor of the arena has changed. And the idea is that if the mouse remembers that this is the novel texture, the one he hasn't seen before, they will spend, because mice have this very well-established novelty preference, he will spend more time on this side of the arena. And this has been, we basically took this from Daisuke Miyamoto in a study published in 2016, showing this to be sleep dependent, showing this task to be sleep dependent and to rely not on the hippocampus, but on M2 and S1 in the cortex. Okay, so we basically do that, and then we have this model that can label the position of the mouse, so we can get a very precise position of even the mouse's nose over the duration of the entire experiment, and we just quantify the exact position of the mouse, at every frame of the video. Okay, and then here's what that looks like. So this is showing the mouse's nose position over the 5 minutes of the recall testing phase. And you can see that on the right, this is the novel side, the right is shown here. He spends more time overall on this novel side of the arena. When he's sleep deprived, that preference basically goes away, as you can see here. And the first thing we did was basically replicate this finding that under control conditions, when the mouse is sleep versus sleep deprived, when the mouse is allowed to sleep versus sleep deprived, in this 1 hour following the acquisition, we replicate that they have a novelty preference if they're allowed to sleep, which is then extinguished if they're sleep deprived.
So it's
and then
not
indeed
quite
we find that when we do the off induction that we are able to rescue that performance.
So with the sleep deprivation, they develop a preference for the familiar side. It's not that it's gone. They develop an anti-preference.
No. So here you can see here that this is not significantly different from no difference, which would be 0.5.
Okay.
So this is showing one example mouse where maybe he does spend a little bit more time on the familiar side, but across all mice there is no difference.
Right. And then the sleep deprivation plus off induction. So then immediately after that off induction, they're placed there.
They're placed. No. So the protocol is that whatever the manipulation is, be it normal control sleep deprivation or sleep deprivation plus off period induction, that only happens in the 1 hour immediately following this acquisition phase. Then another 23 hours elapse, and then the mouse goes back into the arena for the test. So the idea is that this memory consolidation is critically dependent on the sleep in the 1 hour following the acquisition.
I see. So they spend 1 hour in that novel environment, then you take them out, and now they're either sleep, I'm talking about two of the conditions, either they go through sleep deprivation or a sleep deprivation followed by like overlap with an off-period induction.
Overlapped, yep.
Overlap. And now you're saying that, and then after this, so sleep deprivation is how many hours? Like 6 hours?
1 hour.
1 hour of sleep deprivation. And then there is 23 hours during which they can sleep, recover, and all of that.
Exactly.
But that 1 hour of sleep deprivation was sufficient to impair the novelty exploration.
Exactly, and that's just a replication of Miyamoto.
However, if that 1 hour was co-occurring with the both bilateral induction, this is now,
yep.
It's not
here.
Right. Bilateral induction in both M2 and S1.
Right, which again, this paper, we lean sort of heavily on this paper. This paper showed this task to be dependent on M2 and S1.
I see. So you do bilateral induction of on-off states during that 1 hour, and you say that, right. Very interesting. Very, very interesting.
Yeah, so the takeaway here is that normally memory consolidation would be lost to sleep deprivation. But if we do this on-off induction protocol during the sleep deprivation, we're able to rescue that performance back as if the mouse had been asleep.
And another question, do you see a change in the sleep pattern after that 1 hour of sleep deprivation versus after the one that...
Yes, excellent question. So these mice do not have EEGs implanted, so we can't officially sleep score them. However, I did have cameras on all of the home cages. And so we were able to do some rough actigraphy-based estimations. And while it looked like there was maybe a trend happening, that the mice following the off induction might have been less tired, meaning they were less eager to go to sleep following that. There was no statistically significant difference because the variance was just too high and the method can only be so precise.
And what's your explanation of that? Why is it that we don't see a behavioral in terms of sleep pattern readout of
this?
So
it's an interesting question. It could be that so there is work ongoing that's been done and that's ongoing now regarding the role of the cortex in actually regulating sleep behavior and global sleep need. And one sort of idea, I mean, my best idea is just that we're still using relatively low laser powers here. And so my sense is that we're still affecting relatively local cortical networks in M2 and S1. And so most of the brain still is awake. And so the effect that you would see, even if it were there, would be small, and you would probably need a lot of mice to tease that out in a statistically significant way, technically. It could also be that for regulating global sleep need, you want to do this induction pattern in a brain area that is very globally connected, and with a lot of power to influence, other parts of the brain, and so which M2 and S1 certainly may not be suited for. So.
And did you try to see if S1 and M2 are specifically needed for this task? Did you try moving your electrodes, I don't know, to auditory cortex somewhere that you think inducing sleep may not help?
No, we did not. We did not do that, but this reference Miyamoto did. So we basically relied on the pretty extensive characterization that they did, and then replicated the basic finding that the task is sleep dependent, and then just did the experiments in M2 and S1.
Well, it would be very cool if, for example, you see that inducing off period in auditory cortex does not rescue the behavior. So
indeed, yeah.
Okay.
Okay, so then I can just summarize, and then we can discuss anything else sort of anyone would like. So when we induce this non-REM-like on-off activity during wake in local cortical networks during wake, we see basically that this is sufficient to reduce an electrophysiological measure of sleep need, which is slow wave activity. It is able to do that via, we think, reducing electrophysiological measures of neural synchrony, reducing neural synchrony in subsequent sleep, which tonic inhibition cannot do. We are able to fulfill one fundamental homeostatic function of sleep, which is a net synaptic weakening. And we are able to restore memory consolidation otherwise lost to sleep deprivation. And with that said, here are some gratitudes to, obviously, this is a big team effort to everyone in our lab.
Amazing. That was really, really nice. Like the talks on sleep are always so intriguing.
Yeah.
Thanks a lot. Yeah, I wanted to come back to that question of what do you think is about this on and off alternating that is essential to the function of sleep?
Yeah, so this is sort of an open question, a thing we're thinking about, sort of a lot. One idea could be, in sort of all of the previous, we've talked about this before, but we think that this thing, so another sort of fundamental feature of sleep is the sensory disconnection that occurs. Maybe the most striking feature of sleep is that you are completely disconnected from the environment around you. And the reason for that, we think, might be that there needs to be a sort of comprehensive sampling that occurs in sleep to figure out which synapses, which connections, things you need to keep in the brain versus things you need to that are noisy and that need to be down selected, right? Weakened. And so in order to figure that out, you might have to, if you're a neuron or a local cortical network, I'm not sure what scale this might happen over, but if you are some unit in the brain, you might have to sample, go out to all of your possible connection partners and say, basically activate, and then see if that potential partner is coactive with you, meaning that you have a strong or important connection you want to keep, and then keep that one. And then if you basically activate another subset and it doesn't work as well, that might be something that needs to be down selected. And the point is that something like this might be occurring, but if something like that were to be occurring, you must have this thing that you can only get in sleep, where you're disconnected from the environment and where the brain can do its own internal thing. You can't be doing this type of thing when you're constantly being driven by all the lights around you and by questions from your computer and by blah, 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 blah. You must be asleep.
Okay, awesome. Do we have any questions? We're over time and some people left also, but any questions? Okay, I guess we're good. Thank you, Kort. That was awesome.
Yeah. Yes, thank you.
Good luck with the rest of your PhD. And
yes.
so,
Yeah,
have a good day.
thanks. Yeah.
Bye.